Dear ladies and gentlemen, I feel very honored to speak here today. In my topic, I will talk about the effects of high-dose ionizing radiation on microRNA and messenger RNA gene expression alteration in human peripheral blood mononuclear cells. First of all, I would like to thank all those people who helped me to finalize this project. Okay, here you can see a so-called PBMC, a peripheral blood mononuclear cell. PBMCs are in the center of this presentation. Since 2009, it is known that PBMCs that are cultured and irradiated secrete paracrine factors. You can then lyophilize these paracrine factors and use this powder as a medical drug. If you infuse this powder into animals suffering from acute myocardial infarction, these animals show reduced scar formation and improved myocardial infarction, which was published in 2009. To further extend these knowledges, we performed additional experiments in large animals in a domestic pig, and we were able to show that these paracrine factors were able to attenuate scar formation and improved myocardial function in pigs following acute myocardial infarction. You see here histological stainings of heart sections at 24 hours after infarction, and animals treated with APOSEC. APOSEC is the acronym for apoptotic secretome. This is the secretome of these cells. These animals receiving APOSEC showed reduced scar formation. We further performed experiments showing that APOSEC is able to suppress immune activation and immune function and improves myocardial infarctions in a marine model of myocarditis. As you can see here are animals treated with control treatment. You can see a strong influx of leukocytes, whereas APOSEC-treated animals did not evidence a comparable influx of leukocytes. In a further experiment published in 2013, we were able to show that these paracrine factors from stressed leukocytes induce or promulgate angiogenesis and wound healing. You see here pictures of skin biopsies in a marine model of wound healing at day 0 after primary injury. These animals received control medium, whereas these animals received the secretome of apoptotic cells. At day 7 after injury, animals receiving APOSEC had a significantly smaller area of wounding in comparison to control-treated animals. Furthermore, these secretomes augmented the reepithelialization of the wounds and reduced scar formation. Recently, it was published that APOSEC augments or inhibits the ischemic stroke in an experimental model of stroke in rodents. However, we actually do not know what's going on in irradiated or non-irradiated cells during the time period of cultivation. We know that these factors have pleiotrope immune modulatory effects, whereas we do not actually know what biological processes are activated or repressed in these irradiated PBMCs. Therefore, we conducted the following study to first identify biological processes activated or repressed in human peripheral blood mononuclear cells using micro-RNA analysis. Second, we tried to identify messenger RNA, micro-RNA transcription factor interaction networks that would elucidate further biological implications of high-dose ionizing radiation. This slide depicts the study protocol. We first separated PBMC from 4 healthy donors. We then either irradiated or non-irradiated these cells and cultured them for 2, 4, and 20 hours. Thereafter, we separated at the given time points the total RNA content as well as protein, and used these components for further workup. Okay, first we were able to show that ionizing radiation induces apoptosis in PBMC. You see here two electron microscopy pictures of non-irradiated and irradiated PBMCs, and you can clearly see that the cell shape is significantly different between these, let's say, non-apoptotic cells and these irradiated cells, which depicts several hallmarks of apoptosis. For example, the cell fragmentation, the building of apoptotic blobs, the chromosation of the chromatin, and other hallmarks of apoptosis. Furthermore, we performed FACS analysis to elucidate whether typical markers of apoptosis are present in irradiated PBMCs. And we were able to show that irradiated PBMC are positive for annexin and PI, which are both markers for apoptosis or late necrosis. Even at 2 hours after irradiation, we found a significantly higher number of early apoptotic and late apoptotic PBMCs in the irradiated samples. We thereafter questioned, okay, we know ionizing radiation induces apoptosis. Can we find any typical markers that are associated with apoptosis in our samples? We focused on the p53 pathway, as p53 is known to be activated in response to cell stress to apoptosis. We performed a Western blot analysis showing that both p53 and its downstream target p21 are activated in irradiated PBMC, this line. Thereafter we questioned, are caspases involved in the process of high-dose ionizing radiation-induced apoptosis? And we sought to investigate IL-16 as a surrogate parameter for caspase-free activation. There is a nicely published paper showing that the caspase-free activity correlates highly significant with the concentration of IL-16 in the cell supernatant of stressed monocytes. And we were able to identify or to show that at 4 hours after irradiation, the irradiated PBMC released significantly higher amounts of IL-16 in comparison to non-irradiated PBMC, indicating that caspase-free activated cleavage and release of IL-16 is activated in response to irradiation. To further extend this knowledge, we performed membrane arrays investigating 56 proteins associated with apoptosis. You see here a representative membrane array plot of non-irradiated PBMCs, and here is a membrane array of irradiated PBMCs. And it's visible that most detected proteins are detectable in a higher concentration in the samples of irradiated PBMCs. The quantification of these membrane arrays revealed that nearly all investigated proteins were detectable in high concentration in the irradiated sample in comparison to non-irradiated samples. Thereafter, we questioned, okay, we know that all these proteins are activated in response to irradiation. Could we find any other biological processes activated or repressed in our samples? And a way to elucidate this question was to use micro-RNA analysis. And micro-RNA detects the total mRNA content at a single cell suspension at a given time point. By doing so, we first tried to give an answer whether the irradiated sample has sufficient RNA content and an RNA that quality is suitable for micro-RNA analysis. We first performed a gel electrophoresis showing that both the 20S RNA and the 18S RNA were comparable in irradiated and non-irradiated sample, and we were able to prove that the RNA is of good quality. You see only one ladder of these two lines without any degradation ladders. And here you see the electrophoreograms of the same samples, indicating that the 18S RNA and the 20S RNA have one single peak. And here you can see microRNAs with a small nucleotide base pair. Okay, this is the first slide showing results of the microarray analysis of messenger RNAs, the so-called principal component analysis. The principal component analysis is a bioinformatic tool to simplify data of a microarray analysis. One microarray, or one microarray chip, is given as a point in a three-dimensional scale. You can therefore easily get a good overview of your samples, whether they cluster or not, whether they are heterogeneous or homogeneous. What could we see in this slide? We first see that cells separated immediately after blood draw, and the RNA of these cells is clustered on the bottom of the figure, and cells that were cultured for 2, 4 or 20 hours had significantly different messenger RNA expression profiles in comparison to cells separated immediately after blood draw, indicating that the time of cell cultivation changes gene expression profiles. Furthermore, we were able to show that ionizing radiation induces changes of global gene expression. As you can see here, PBMCs irradiated and cultured for 2 hours, for 4 hours, and for 20 hours. Thereafter, we tested for differentially expressed genes between irradiated and non-irradiated PBMC, and we were able to see that several genes are upregulated in a time-dependent manner, whereas at 2 hours after irradiation, only approximately 160 genes were upregulated. At 20 hours after irradiation, more than 2,800 genes were upregulated in irradiated PBMC, indicating that induction of apoptosis causes the expression of genes, that it is an active enforced process. Furthermore, we were able to show that several other genes were down-regulated at the same time, also showing a time-dependent increase of differentially expressed genes. If we focus on these 5 and 25, or these 30 genes, and displaying these genes in a heat map, you can see that we have two big different clusters. One cluster, this red one, is upregulated in non-irradiated cells in a time-dependent manner, whereas this red cluster is upregulated in irradiated PBMCs, whereas non-irradiated PBMCs did not express these genes in a comparable manner. To further elucidate the molecular or biological function of these proteins, we used canonical pathway analysis, named the KEC pathway database. The KEC database is a publicly available database which clusters genes based on their molecular function and their involvement in different pathways. And we were able to see that the upregulated genes in irradiated PBMCs are associated with the p53 signaling pathway, with the mRNA surveillance pathway, with pathways associated with cancer, and more interestingly, with the lysosome, the phagosome, with the chemokine signaling pathway, and with a pathway named adherence junctions. We found several genes upregulated coding for proteins involved in the actin-myosin polymerization, in the polymerization of the cytoskeleton. We also performed quantitative real-time PCR validation of the microarray data. In this slide you can see a selected set of genes that were validated with qPCR, and we were able to find a highly significant correlation between the microarray data and the qPCR data, indicating that the microarray data is valid and we can use these data for our bioinformatic analysis. To get a broader overview of higher regulatory motifs that might be regulated or that might be responsible for the gene expression changes in messenger RNAs, we questioned whether transcription factors might be activated in response to ionizing radiation. A transcription factor is a protein that activates co-activating factors and polymerizes with the DNA polymerase II. And this complex then activates the transcription of DNA into RNA. And we were able to see by using bioinformatic tools that at 2 hours after irradiation, only one transcription factor should be activated based on the bioinformatic analysis, and this was P53. P53 should be activated at 2 hours after irradiation, whereas at 4 hours after irradiation or at 20 hours after irradiation, P53 was not identified as a significant or as an important transcription factor, whereas this one, Krüppel-like factor 4 or SP1 or this zinc finger protein, had been shown to be activated in response to irradiation. We first checked whether these transcription factors are really present on mRNA level. Here you can see a plot where we were able to evidence that all 4 transcription factors are present on mRNA as well as on protein level, as you can see here in this immunoblot analysis. Therefore, we were able to validate that it might be possible that these transcription factors are involved in the regulation of ionizing radiation-induced biological changes. What is known about these transcription factors? It's well known that Krüppel-like factor 4 is a downstream target of P53. This is in line with our data, where we were able to show that at 2 hours P53 is activated, whereas at 4 or 20 hours only Krüppel-like factor 4 is activated. In addition, it's known that SP1 or this zinc finger protein are involved in the execution of apoptosis, and therefore we can provide further evidence for their involvement in apoptotic signaling pathways. Thereafter, we focused on micro-RNAs. Micro-RNAs are small, non-coding RNA transcripts that are processed in the cell nucleolus and released into the cytosol. In the cytosol, they can either alter the messenger RNA translation or even inhibit or degrade messenger RNA. Therefore, a micro-RNA targets a messenger RNA and inhibits the translation of the messenger RNA. Therefore, less protein is produced by these messenger RNAs. And we focused on the micro-RNAs visible or detectable with our Agilent chip. And you see here a heat map of all expressed micro-RNAs in our selected samples. And what can you see is that PBMCs at 20 hours after irradiation express the highest amount of micro-RNAs. These are micro-RNAs that are highly expressed only in irradiated samples at 20 hours after irradiation, whereas this cluster of micro-RNAs is repressed in irradiated samples. If we take a closer look about these micro-RNAs, you see that, for example, the micro-RNA 887 is upregulated in irradiated PBMCs with a fold change of 8 to 7, which is pretty high for micro-RNA alterations. We found several other micro-RNAs that have not been described in literature so far. We also performed qPCR analysis of these microarrays, showing that we have a strong correlation between chip data and qPCR data, as you can see here for MIR 8817, which was also upregulated with an increase of 7 in the qPCR. We then questioned, okay, can we find any negative correlation pairs between microRNAs and messenger RNAs? This slide depicts microRNAs, given here in green or red, and their corresponding downregulated messenger RNAs. What can we see in this slide? It's pretty simple. Several microRNAs target many messenger RNAs, and we find a close interaction network between upregulated microRNAs and downregulated messenger RNAs. To further extend our knowledge between microRNAs, messenger RNAs, and also transcription factors, we performed a so-called networking using a bioinformatic tool called MAGIA2. MAGIA calculates first a target of a microRNA, named messenger RNA, and targets of messenger RNAs, also transcription factors. And it also incorporates information whether a transcription factor induces the expression of messenger RNAs. Therefore, we get highly complex interaction networks between these three types of genetic informations. What did we get when we use our data? We found that one protein or one transcription factor is highly significantly involved in the regulation of microRNA-induced alteration. Its name is hepatic leukemia factor. Hepatic leukemia factor is a central hoop node which is downregulated in response to ionizing radiation. It's targeted by multiple microRNAs which were all upregulated in response to irradiation. And these microRNAs, on the one hand, target HLF, which is downregulated, and simultaneously target several messenger RNAs, which were also downregulated. Furthermore, these factors should upregulate these messenger RNAs. However, it is downregulated, therefore he cannot activate the transcription of these factors. We validated our bioinformatics data, showing that HLF was significantly downregulated on qPCR with qPCR, and we also performed immunoblot analysis showing that irradiated samples have less protein content of HLF in comparison to non-irradiated sample, as you can see here in the figure. To our knowledge, there is only one publication dealing with the protein HLF and the process of apoptosis or cell death, and the authors were able to show that an ectopic expression of HLF was able to attenuate apoptotic processes. Therefore, it's in line that during apoptosis, HLF is downregulated or repressed and should therefore enforce the execution of apoptosis. Finally, we performed micro-RNA fishing. Micro-RNA fishing is a method to identify a single microRNA that might be responsible for the genetic expression alteration of messenger RNAs. What can we see in this slide is that we cannot identify one single microRNA. Instead, we found several microRNAs that might be involved in the regulation of transcriptomic expression alterations. We see here several different nucleotide base pairs that are involved in the regulation of messenger RNAs. To sum it up, what did this study show? First, we were able to show that high-dose ionizing radiation induces apoptosis in human PBMC. Then we were able to show that both messenger RNAs and microRNAs are regulated in response to irradiation. The highest expression alterations were visible at 20 hours after irradiation in PBMC. We identified 4 transcription factors that might be involved in the regulation of apoptotic pathways. And we found that the transcription factor, hepatic leukemia factor, is a target of several upregulated microRNAs and itself is downregulated in irradiated cells. We conclude that the induction of apoptosis by ionizing radiation is an actively enforced process that is regulated by microRNAs, transcription factors, and messenger RNAs. Further studies are warranted to elucidate transcriptomic interactions between microRNAs, messenger RNAs, and further regulatory events. Thank you for your attention.